Assalamu alaikum students, hoping you all will be fine. Today we will discuss about using the recombinant DNA technologies to detect disease allele directly. हम आज ये देखेंगे कि हम रिकम्बिनेंट डीएनए टेक्नोलॉजीज़ को किस तरीके से यूज़ कर सकते हैं ताकि हम कोई भी डिजीज़ है जो जेनेटिक है हम उसका जो अलील डायरेक्टली रिलेट कर रहा है विद दैट डिजीज़ को डिटेक्ट कर सकें. A recessive mutant phenotype that follows single gene inheritance are responsible for more than 500 human genetic disorders. Around 500 human genetic disorders are related with the mutant genes. A homozygous person resulting from a marriage between two heterozygous for the same recessive allele will be affected by the disease. We people know, say for example, if we establish a cross between the two heterozygous individuals, here we can see this is capital A, small a, and marriage if established with the heterozygous individual, both of these will produce two types of gametes. Yeah, this one again, capital A and with small a. So because of the fusion of these two, the homozygous recessive individual will be produced. This individual will be affected. Cells derived from the fetus can be screened at an early enough stages to allow the option of abortion of afflicted fetus. If we people get information that this coming baby is either affected or not, on the basis of this information, we can decide whether to hold this baby or not, whether to carry this pregnancy or to abort it. And but it could only be happen if we have any information regarding the coming fetus or coming baby will be affected or not. Among such kind of these techniques, the amniocentesis is the first procedure. To detect such genetic defects, fetal cells can be taken from the amniotic fluid. From the amniotic fluid, we take fetal cells. Then we separate for then from this separated from the other components because when we take the fluid, uh, many of the other components will also be present in that in that fluid. So at that time we need to separate these cells because these cells contain amniotic fluid as well as the cells. We will centrifuge it, and on this centrifusion fusions the cell will be settled down. Then we culture these cells on the culturing media, on the whatever the media is required. On the culturing media, we will culture these cells. After the culturing of the cells, the analysis of chromosomes, protein, enzymatic reactions, and other biochemical properties can be checked. This process is known as the amniocentesis because on the basis of amniotic fluid we are performing this process. That's why this process is known as the amniocentesis and can identify a number of known disorders. On the basis of this study, on the basis of this amniocentesis procedure, we use to detect whether the chromosomes are performing normal function or abnormal, or whether the number of chromosome is normal or abnormal. So, for example, chorionic villus sampling (CVS) is a related technique in which uh, a small sample of cells from the placenta is aspired uh, by using a long syringe. CVS can be performed earlier in the pregnancy than than can amniotic synthesis. CVS is also like CVS is also the sampling technique, uh, but it can be performed before early as compared to the amniocentesis, which must await the development of the large enough volume of the amniotic fluid. In amniocentesis, we need to wait that amniotic fluid develop and it will be available in the large quantity. Then we will be able to get the sample. But chorionic villus sampling is the sampling from the placenta, which does not require the development of the amniotic fluid. If these are the diseases and their approximate incident among the live births, that like the cystic fibrosis, one in among 1600 Caucasians, muscular dystrophy, one in 300 boys, X-linked disease, Gaucher disease, one in 2500, etc., etc. Whatever the sickle cell anemia, you can see one in 400 US black in some West African population, and so on and so forth. Alteration of the restriction sites by the mutation. Sometimes we can create the mutations to cure the diseases. Sickle cell anemia is genetic disease that is caused by the well-characterized mutational alteration. We people know about the what kind of mutation is take place in this disease, and we know that affecting approximately that this disease affect approximately 0.25% of the African Americans. The disease is actually the because of the defect in, or we can say the alteration in the hemoglobin, in which the amino acid valine substitute for the glutamic acid at a position 6 in the beta globin chain. The change in the sequence in the DNA is GAG to GTG. Here you can see only a single base, only a single base. Because of only a single base, this disease is being caused. So because of this, by using the restriction enzymes, we can detect uh, whether this defect is genetic or not. With that, what kind of organism, what kind of uh, we can see what kind of mutation is present over there. GAG is being converted into GTG. The change eliminates a cleavage site for the restriction enzyme MST2, which cuts the sequence CCTNAGG, in which N represent any of the four bases and is representing any of the four bases. The change from CCTGA, CCTGA, GG to CCTGTGG. Here you can see this is actually the change CCTGTGG and CCTGAGG. Here GAGG and here the GTGG. You can see this is actually the difference because of with the disease sickle cell anemia is being caused. Okay. This can also be recognized by using the Southern analysis with the use of the labeled beta globin cDNA as a probe, because the DNA derived from persons with sickle cell disease lacks one fragment contained in the DNA of the normal person and contain a large fragment not seen in the normal DNA. This is actually happening in this in this process in this disease. Here you can see this CCT creates proline, GAG creates glutamine, GAG creates glutamine here. So in the diseased, so for example, is the disease in the disease sickle cell anemic patient, CCT creating proline, GTG creating valine, and GAG is creating glutamine. Here is only a single change, only a single change, which is converting glutamic acid into the valine. This is not glutamine, glutamic acid. Glutamic acid into the valine. So this site for the glutamic acid can be chopped out, can be detected by treating it with the MS2 enzyme, restriction enzyme. We can detect it on the gel electrophoresis, and from there we can make a southern blot for this. Probing for altered sequence when a genetic disorder can be attributed to a change in a specific nucleotide, synthetic oligonucleotide probe can identify that change. We can design the specific probe to identify such kind of genetic changes. The best example is the alpha antitrypsin deficiency. Alpha antitrypsin deficiency. Because of this uh, deficiency, a great increased probability of developing pulmonary emphysema is developed. I must say, because of this deficiency, pulmonary emphysema is developed. The condition resulting from a single base change at a non-nucleotide position. Synthetic oligonucleotide probe that contains the wild type sequence in the relevant region of the gene can be used in a southern blot analysis to determine whether the DNA contains the wild type or the mutant sequence. At a higher temperature, a complementary sequence will hybridize, whereas a sequence containing even a single mismatch base will not match. Okay, for this purpose, I have the diagram. For this purpose, say for example, we know that on this position, this is the sequence. This is complete sequence, and we know this. Okay, just a minute. So for example, this is the sequence, and we know here the change is here the change may happen, may occur. Here the change may occur on this position. We design a probe that will match. So for example, this is the wild type. This is wild, and this is the DNA which is muted. Here is here is the change may occur. Okay, this is the mutant. We design the probe. Probe will match over there. Will will match over there, and for this it will not. Okay. For this, there is no binding site for this one. So from this probing, we can identify whether our required gene is present or altered gene, whether our required DNA sequence is present or altered DNA sequence is present. PCR test, we know about the PCR test, large number of PCR tests for the different genetic as well as pathogenic diseases are available, because PCR allow an investigation to zero in a potentially defective DNA sequence. It can be used effectively in diagnostics of diseases in which a specific mutation site is in question. Primers spanning the site are used. We use such kind of primer. We use we can use the primer specific to the specific sites. DNA can be isolated and sequenced or otherwise compared with the wild type. We have already converted the detection of linked RFLPs as a more indirect diagnosis of the presence of diseased alleles. Gene therapy, the next is the treatment for the genetic disorders, gene therapy. The general approach of gene therapy is nothing more than an extension of the techniques of the clone selection by the functional complementation. The functions absent in the recipient as a result of defective genes are introduced on a vector that insert into one of the recipient chromosome, and thereby generates a transgenic animal that has been genetically cured. In this technique, say for example, an individual is carrying a defective part of the DNA, and say for example, this is the defective part of the DNA. By using the vectors, we introduce the potential active part of the DNA in this defective region. This will bind over there and perform the required function. However, gene therapy is also being applied to mammals other than the human. Uh, Up till now, mostly um, It's being used for the mammals. First example of the gene therapy in the mammal was correction of the growth hormone deficiency in the mice. In the mice, the recessive mutation little result in dwarf mice. Because of the deficiency of the growth hormone, the dwarf mice are generated, even though mouse growth hormone gene is present and apparently normal. No mRNA for this gene is produced. The initial step in correcting this deficiency was to inject homozygous little egg with about 5,000 copies of a 5 kb linear DNA fragment that contained the rat growth hormone structural gene. And this rat growth hormone is fused to a regulator promoter and sequence from a mouse metallothionein gene (MP). So for this, we combine the rat gene and regulator promoter of the mouse gene, uh, metallothionein gene. Normal job of the metallothionein is to detoxify heavy metal. This is required for the detoxification of the heavy metals, but here we are altering its sequence. We are just using its promoter. The eggs were then implanted into the pseudopregnant mice, and about 1% of these baby, and the mice were, and, uh, and the pregnancy was led to the birth of the organism. The eggs were then implanted into the pseudo pregnant mice, and the baby mice were raised. About 1% of these babies turned out to transgenic. 1% transgenics are produced, showing increased size when heavy metals were administered in the course of development. A representative transgenic mouse was then crossed with a homozygous little female. Representative transgenic mouse was crossed with the homozygous female. And then the pedigree is shown. Then the pedigree is shown that can be seen in the figure that the mice 2 to 3 times the weight of their little relatives are produced in subsequent generations with the rat growth hormone acting as a dominant allele, always heterozygous in this pedigree. The rat growth hormone transgene also make it positive mice bigger. If you can see, this is the gene sequence, and this is the MP region, this is the rat growth hormone, and the construct is made and injected into the homozygous recessive female egg, and then this is transplanted into the um, pseudo pregnant mouse. From here, the transfer from here. Uh, the organisms are raised and their pedigrees are developed, and after their analysis we found that there is the trans after the analysis we found that most of the large weight organisms are transgenic. The rat growth hormone under the control of the mouse promoter region that is responsive to the heavy metal is inserted into a plasmid and used to produce a transgenic mouse. RGH compensates for the inherent dwarfism in the mouse. RGH is inherited in the Mendelian dominant pattern in the mouse pedigree. Now we're going toward the human gene therapy. Um, like the mouse, human gene therapy uh, is also almost the same process. Perhaps the most exciting and the controversial application of the transgenic technology is the human. This is very important to know that not only in the Muslim world, uh, in non-Muslim world, the use of this technology is also under debate. Whether we must use this for the human or not. The treatment and the alleviation of the human genetic diseases by adding exogenous wild-type gene to correct the defective function of the mutations. We have the treatment and the alleviation of the human genetic disease by adding exogenous wild type genes to correct the defective function of the mutations. We have seen that the first case of gene therapy in mammal was to cure genotypically dwarfed fertilized mouse egg by injecting the appropriate wild type allele for normal growth. The like that we have studied before these slides. This technique has a little application in the humans because it is currently impossible to diagnose whether a fertilized egg cell carries a defective genotype without without destroying the cells. However, in an early embryonic containing only a few cells, one cells can be removed and analyzed with no ill effect on the remainders. This is the technique that we used earlier. I told you that for the mouse, there say for example, these are the two mouse. We get the cells from this one, and then integrate the cells of this white mouse with this one, and then inject it into the pseudo mother. After getting the babies, we will get the chimera, and then we will cross the chimera with the light colored mouse, and after their cross, we will get the dark colored mouse, which will be probably transgenic. Type of the gene therapy. Types of the gene therapy. Um, there are almost three types of the gene therapy. One is injecting the fertilized egg. Number two is the germinal therapy, and number three is the somatic therapy. Uh, in you can say in injecting the injecting the fertilized egg, we take our gene of interest and inject it into the egg of the mouse. Germinal therapy, we take the transgenic cell and insert it into the blastocyst stage, which will make the mosaic gonads, whatever the mosaic requirement, whatever the requirement, whether to make the somatic mosaic cells or whether to make the gonadal mosaic cells. Somatic therapy, we take the genes, the transgenes, and insert it into the organism. We take the cells and make it transgene and insert into the organism, which will lead to the transgenic clone. The basic type of the gene therapy can be applied to the human germline and somatic cells. The goal of the gene therapy is the more ambitious to introduce the transgenic cell into the germ line as well as into the somatic cell population. Not only should this therapy achieve a cure of the person treated, but some gametes could also carry the corrected genotypes. We have seen that such germinal therapy has been achieved by injecting mice cells. However, the protocol that is relevant for the application of the human is the removal of an early embryo with the defective genotype from a pregnant mouse and inject with the transgenic cells containing the wild type allele. These cells become part of many tissues of the body, often including the germline, which will give rise to the gonads. Then genes can be passed on to some or all progeny depending on the size of the clone of transgenic cells that lodges in the germinal area. However, no human germline gene therapy has been performed to date. Somatic gene therapy, therapy is not done up to the day. Focus only on the body, soma cells. The approach is to attempt to correct a disease phenotype by treating somatic cells in the affected person. At present, it is not possible to render an entire body transgenic. The entire body cannot be transgenic. In such cases, it is likely that not all the cells of dead tissues need to become transgenic. A percentage of cells being transgenic can ameliorate the overall disease symptoms. The method proceeds by removing some cells from patient with the defective genotype and making these cells transgenic through the introduction of copies of the clone wild type genes. The transgenic cells are then reintroduced into the patient's body where they provide normal gene function. Explaining. In this method, we remove some cells from the patients which have defective genotype. Then we transgene these cells. Then after removing here, you can see we remove we remove the cells from the this organism and make it transgenic. After transgenic, again inserted into this organism. Again inserted into this organism. Just a minute. Then the cells are again inserted into the in that organism. These transgenic cells are then reintroduced into the patient's body, uh, where they can provide the normal gene function. Our next topic is the cancer. We will discuss in with regards to the cancer mutations categories, ability to proliferate, increase, decrease in apoptosis, oncogene mutation, and mutation in suppressor. Have tumor promoting mutations been identified? Modern pedigree analysis we can use in modern pedigree analysis, we can use the cytogenetic analysis. With the familiar tendency towards certain kind of cancer can be mapped relative to the molecular markers, such as we can use the microsatellite markers, and in several cases this mapping has led to the successful identification of the mutated gene. We can detect by using the microsatellite or the pedigree analysis. Uh, um, um, Pedigree analysis can be used to detect the mutated genes. Cytogenetic analysis, tumor cells themselves are also has proved invaluable. We can use the tumor cells to identify what kind of problem they have. Many types of the tumors are typified by characteristic chromosomal translocations or because of the deletions of the particular chromosomal region. In some cases, these chromosomal rearrangements are so reliably a part of particular cancer that they can be used for diagnostic purpose. We can say that we can analyze the tumor cells and can we can karyotype these cells to find out whether there are deletions or whether there are defects in the chromosome. What kind of defects in the chromosome? There are three types of the oncogene mutations. Oncogenes may have point mutation, they have loss of protein domains, and they may have the gene fusions. In the point mutation, there could be the point mutation. So, for example, if we recall the normal RS protein, G protein subunit that takes part in signal transduction and normally function by cycling between the active GTP bounded state and the inactive GDP bound state. In point mutation, only a mutation on a single point can lead to the oncogenesis, can lead to the cancer generation. In point mutation, I told you that, say for example, a G protein which work by cycling between the active GTP and the bounded GDP forms. Because of this point mutation, this cycling is disturbed. Loss of protein function. Some protein function is lost because of the mutation in the genetics, because of mutation in the chromosome. Say, for example, structural alteration can also be due to the deletion of part of a protein. The ERBB oncogene encodes a mutated form of an RTK known as the EGFR a receptor for epidermal growth factor ligand. Then the fusion, the genes may fuse with each other and the function of the gene may disturb. The classic example of fused gene emerged from studies of the Philadelphia chromosome, which, as already mentioned, is a translocation between. There is a translocation. Fusion means the two parts are fused. So there could be a transfusion. There must be a translocation between chromosome 9 and chromosome number 22. That is diagnostic feature of the chorionic myelogenous leukemia (CML), Cronus myelogenous leukemia. Recombinant DNA methods have shown that the breakpoints of the Philadelphia chromosome translocation in different CML patients are quite similar and cause the fusion of two genes, BRCA1 and ABL. So in this disease, uh, two genes are fused because of the translocation. Two genes are fused, BRCA1 and the ABL. Inheritance of the tumor phenotypes. So, for example, we discuss about the retinoblastoma. We need to know that RB genes of the cell cycles are mutated, arise in the retinal somatic cells. Uh, how and retinoblastoma, RB genes of the cell cycle are mutated and arise in retinal somatic cells. Uh, the RB protein function by binding of the E2F transcription factor. Bounded RB prevents E2F from promoting the transcription, the protein prevalent DNA replication. Retinoblastoma disease में RB genes are required for cell cycle cell cycles. RB genes जो हैं वो सेल साइकिल के लिए रिक्वायर्ड होते हैं. इन इसमें RB genes which are required for this cell cycles इनमें mutations हो जाती है और उन सेल्स में म्यूटेशन हो जाती है विच जिनका काम ये है कि वो रेटिनल सोमैटिक सेल्स को प्रोड्यूस करते हैं. इसमें होता ये है कि आरबी प्रोटीन जिसका काम ये है कि वो बाइंड करे E2F ट्रांसक्रिप्शन फैक्टर के साथ. अब E2F ट्रांसक्रिप्शन फैक्टर का क्या काम है? आरबी प्रोटीन E2F ट्रांसक्रिप्शन फैक्टर के साथ बाइंड कर रही है. बाउंडेड आरबी अगर आरबी E2F फैक्टर के साथ बाइंड करती है तो ये जो इसका काम ये होगा कि ये E2F को ट्रांसक्रिप्शन जो है प्रोटीन की ट्रांसक्रिप्शन करने से मीन्स जस्ट अ मिनट. ओके. आरबी जीन्स सेल साइकिल में म्यूटेट हो जाते हैं अ, उन सेल्स के अंदर जिनका काम ये है कि वो रेटिनल सोमैटिक सेल्स बनाते हैं. अब यहाँ पर होता ये है कि आरबी जीन का ओरिजिनल फंक्शन ये है कि ये E2F ट्रांसक्रिप्शन फैक्टर के साथ बाइंड करते हैं. आरबी जीन जब E2F ट्रांसक्रिप्शन फैक्टर के साथ बाइंड करते हैं तो ये जो E2F है इनके फंक्शन को डिस्टर्ब करते हैं ताकि वो फर्दर ट्रांसक्रिप्शन ना कर पाए. ये जो ट्रांसक्रिप्शन अगर E2F फैक्टर्स करते हैं इनकी इस जीन की ट्रांसक्रिप्शन होती है तो जो प्रोटीन बनेगी वो डीएनए की रेप्लिकेशन में हेल्पफुल होती है. डीएनए को रेप्लिकेट करवाती हैं. लेकिन जब आरबी जीन E2F के साथ बाइंड कर जाती है तो वो उसको ट्रांसक्राइब नहीं होने देती जिसकी वजह से डीएनए की रेप्लिकेशन नहीं हो पाती. अगर एक एक्टिव आरबी इसके साथ E2F के साथ बाइंड ना करे तो E2F जो है वो ट्रांसक्रिप्शन जो एस्पेस की ट्रांसक्रिप्शन है उसको चलाता रहेगा और डीएनए की रेप्लिकेशन होती रहेगी. तो इस वजह से E2F will remain always in the S phase. डीएनए की रेप्लिकेशन होती रहेगी बिकॉज डीएनए की रेप्लिकेशन हो रही है सेल्स की ग्रोथ होती रहेगी. Then how to resolve this issue? How to resolve this issue? How we can resolve this issue? After there is no solution for there is no solution. Gene therapy may be effective. Next tumor gene is the p53 tumor suppressive gene, a link between the cell cycle and the apoptosis. Another very important recessive tumor promoting mutation, uh, which is the p53 gene as a tumor suppressor gene. This gene is actually responsible to develop the uh, cancer. The active p53 protein is transcriptional regulator that is activated in response to the DNA damage. Activated wild type p53 serves double duty. It has a double function. Function number one, it prevents the progression of the cell cycle until the DNA damage is repaired. And under some circumstances, it induces apoptosis. In the absence of functional p53, the p53 apoptosis pathway does not become activated. If the p53 is mutated, the apoptosis cannot be happen, and the cell cycle progress even in the absence of the DNA repair. This is all about the cancer. I hope I must stop over there. Um, Today we have learned about the two genes regarding the development of the cancer. Number one is the p53 gene, which is the retinoblastoma gene, which are defect on which may lead towards the cancer development. Thank you, students. Hoping your day will be fine. Good luck.